Hello viewers, my name is Damilari. In this lesson, we're looking at the topic the cell and its environment. The cell and its environment. When you hear cell, that's the unit of life. It means as we are, we have cells, and that's why we are alive. So the cells in us, we have to consider their environment if they will last, if they will not die easily, if they will not malfunction. And what are the environments? Water. It means the water where the cell is found, hope is not polluted. Air, soil, the parts of living organisms where we can find cell or cell in another cell or cell in a tissue. How is the environment? So it means if the environment is not polluted, if the environment is not having too much concentration that can affect it, then it will survive. So, and that's why we have to look at the importance of the cell's environment. The first one, we have food. It means it is from the environment that cell can get their food. Likewise, oxygen, they can get oxygen from the environment. It is from the environment that cell can become moist. That means having liquid in their body so that they can be alive, so that they will not dry up. It is from the environment they can excrete, remove waste products. From the environment they can gain their protection. That's buffer zone. It is from the environment they can carry out chemical reaction, reacting with the environment. What are the materials cells can exchange with their environment? Food can be exchanged. They can either get food from their environment or release food to the environment. Likewise, gas, talking about oxygen or carbon dioxide. It is from the environment they can exchange waste, excretory substance, or water. So these are the materials that can be exchanged with the environment. Now we move to biophysical processes. When you hear biophysical processes, it involves exchange of materials. Biophysical process, it means, bio means life, means that our living organism will communicate with the environment by exchanging certain materials. So these processes include diffusion, osmosis, active transport, plasmolysis, turgidity, hemolysis, and crenation. Let's start with diffusion. When we talk of diffusion, what kind of exchange is it talking about? This one has to do with how solid or liquid or gas, how they will be moving randomly from the region of higher concentration to the region of lower concentration. It means liquid can move, solid can move, gas can move. And if they will move, there's something that will aid them, that will help them. That means the medium. That means air can make gas to move. Liquid can make liquid to move, including solid. So it means that movement now through this air, liquid, and so on, if the movement is coming from a higher concentration to a lower concentration, you will say there is diffusion, and that will continue until there is uniformity. For example, when you look at dead rats, there is a kind of foul smell coming from it. So that dead rat now is having higher concentration. So the smell moves into the environment through air. That's the medium. So it means the smell will now be diffused. The same thing, looking at air freshener, it has a pleasant smell. So it means from the high concentration of the air freshener to the environment, the air, where we have low concentration, there will be a kind of diffusion and everyone begin to perceive the smell. So mean that it is now uniform. The smell is not only in the container of the air freshener, but is now spread out. That's what we call uniform. It is now uniform everywhere. The same thing, ink, that one now is liquid. When you pour it in water, it also spread until it covers the whole water. So that's diffusion. What are the factors that can affect diffusion, that can make all these things spread very fast? Surface area. When the area is very large, it means it will spread easily. Just like when you are in a large room, you can play as you like. Temperature, if the temperature is very high, it means it will help that diffusion to make the liquid or the air or the gas spread very fast. Size of molecules, molecules that are very small, they will diffuse very fast, just like small stone can go far when you throw it. Presence of concentration gradients, those substances that have high concentration, they will diffuse very fast, they will spread very fast. Then state or nature, talking about, is it gas, liquid or solid? Something that is a gaseous substance will diffuse very fast. Shaking or stirring, when you shake substance, it means what the content of that substance will easily spread. You will easily perceive as you are shaking, you will know something is smelling somewhere. Now, what is now the importance of diffusion to living things, since biologists do it live? So why do we need diffusion? You can use this mnemonics: problem must die by fire. It means you take letter P, those ones that are in blue ink. Problem P, must, M, S and T. Die, D, fire, by fire, FIRE. The letter P, the importance of diffusion is that it will help photosynthesis take place. That means for photosynthesis to take place, there must be diffusion. That means gas must diffuse. And what is that gas? Carbon dioxide coming into the plants. That's for P, photosynthesis. M, that's minerals. For the, minerals movements into the body of plants is also diffusion. You know, plants needs mineral resources that make them survive. So that's the mineral will diffuse into the body of that plant. Then S. S has to do with saprophytes. Saprophyte, talking about those organisms that take that get their nutrients from dead organisms. So it means the movement of that materials or of the nutrients into the body of that organism is diffusion. It means the food is digested already, so it's just for it to enter into the organism. So that entering is called diffusion. So it means diffusion of digested food into the body of organism. Then the next one, F. I'm following this mnemonic. So for F now, we can pick the word fetus. Fetus means baby in the womb. So baby also, there must be diffusion. Food must be diffused into the body of the fetus from the mother. Then I. I is intestine. It means from the intestine there must be an absorption of digested food. Food that is digested must be diffused into the intestine. That's intestinal wall. R, that's talking about respiration. It means it is through respiration that we notice that there is diffusion. When you are respiring, you are breathing in oxygen, breathing out carbon dioxide. It means there is a diffusion of air in that person's body. Then E, excretion. You diffuse out waste products. So that's for the importance of diffusion. The next thing we look at now is osmosis, another biophysical process. Osmosis, this one now is just opposite of diffusion, meaning that this one will be moving from lower concentration to higher concentration. And this one will have to involve a semi-permeable membrane. That means you must permit the movement of liquid. But in diffusion, that one doesn't need any permission. You don't need semi-permeable membrane. So, and this one, its own concentration, there must be a change or difference in concentration. There must be low and high before osmosis can occur. But diffusion, it's not until there is low and high. So it will take place in any place. So now let's look at osmosis. We said movement of water, only water, take note, not solid, liquid and gas. So only water will have to move, and it must be through semi-permeable membrane. Then it will be from lower to higher concentration, and until there is equilibrium. That means it will keep moving from lower to higher until it is equilibrium. And when I say it's equilibrium, it means no movement again. Now, the solution which loses water in osmosis, we call it hypotonic. It means when there is an hypotonic solution, it means it's a weak solution. It means water can easily be lost. So in hypotonic, so since it's not powerful, but hypertonic, it will be gaining. You know, hyper is just like someone that is very hungry, so it will be demanding for water, or thirsty, demanding water. But hypo, so it means is enough, so it doesn't need to demand for water. It would rather be giving out water. That's why hypo is losing water. So the equilibrium state is called isotonic state. That means it's balanced. So it doesn't need to demand for water. It doesn't need to lose water. It's just at the neutral state, and that's isotonic. So no gain or loss of water. So this diagram explains osmosis. Look at these big circles representing big solutes. It means when the concentration is high, we have high salt content, that's for this big, and low water. But this other one, it means we have low salt and high water. So meaning that this place where there is much salt, bigger salt, high concentration of salt, it will be demanding water, and that's why look at this arrow telling us that water will begin to move to this region. So meaning that there is movement from lower to region of higher concentration. This line is through the semi-permeable membrane. Now the next thing we look at is the word osmotic pressure. Anywhere there is osmosis, there is a pressure, something pressurizing the flow of that water. So meaning if the environment is like environment of a distilled water, there is no special pressure. So that's why the osmotic pressure in distilled water is 0. It means there is no high concentration there, so it's 0. But if the environment is higher amount of solute that is dissolved in solution, there will be high osmotic pressure. Just like when you take a peppery food, too much pepper, you begin to demand for water. So that water demand is what we call high osmotic pressure. So pressure looking for water. Now from the above solutions now, that means what I've just explained, osmotic pressure we can now define as the pressure that is developed by a more concentrated solution. So it means if the solution is concentrated, that means there's too much salt, too much sugar, it means you need to expect water. There will be a kind of pressure. So now and since there will be demand of water, so water will begin to move into it through a semi-permeable membrane. So and it can also be defined as the pressure that we require to stop water. Remember that the water was being demanded because of the high concentration, so absorbing. So if it has absorbed, then it can also cease to release water. It means if you stop water from diffusing, you will say, oh, the water have grabbed, I must not let it go. Just like in the desert, how they conserve water. So the pressure with which they got the water is also the pressure with which they will retain the water. They will stop the water from diffusing. So therefore, osmosis also, we can still define it as what? Movement of water molecules from lower osmotic pressure to higher osmotic pressure. That means the place where there is no much disturbance, so water will move to the place where there is much disturbance, high osmotic pressure, but it must be through semi-permeable membrane. What is the meaning of that semi-permeable? From the word permeable, it can allow tiny pores. That means it has tiny pores, tiny holes that will permit movement of small molecule, and that molecule is water. So not gas, not solid. So majorly water. It will allow it to move. So and now since it's allowing the water to move, as the water is being drawn, as it's moving from into the solution A, the one that is very strong from that diagram. So if it's moving into solution A, coming from solution B where there is no much concentration, so that osmotic pressure of A will be slowly reduced. That means that demand is just like someone that is very thirsty. By the time he starts drinking water, the thirst will be reducing. So that's how it operates. Now until, and that will continue, that means it will be reducing until it is now at in an equal form, equal equilibrium. B, there was movement of water from B to A, so it will keep moving. The demand will be going down, demand will be going until it doesn't need water again. It means they are now in equilibrium state, and that's what we call osmotic equilibrium. So the rate of the movement of water from A to B, so is equal to the rate of movement from B to A. That means once it has gotten to osmotic equilibrium state. Now what are now the importances of osmosis? The way I use the mnemonic for diffusion, that problem must die by fire. So this one for osmosis, just say charts, king, cross. That means you can get it from the word osmosis, like Moses in the Bible. So he had to chart the king Nebuchadnezzar. So he had to chart the king that, okay, we need to leave this place and cross to the other side. Sorry, I said Nebuchadnezzar, that was Pharaoh. So you have to chart Pharaoh, the king, that the people of Israel need to cross to the other side. So that's C. What does it mean? That's cell. It means osmosis will allow water to move from one cell to another. That's C. H, you remember air of roots. So osmosis will allow water to move or to be taken up, so through the root air. So it will allow water to be taken up by the root air. Then this A is not part of it, but we move to T. T is turgidity. It means osmosis will allow turgidity. It means make plants to get soaked with water. It will absorb the water until it's turgid. So then S, that's stomata. Plants, they have stomata for respiration. So that stomata always open and close for gas to be exchanged. So it means osmosis will make the stomata to be opening and closing so that it can allow gas to be exchanged. K, use it to remember kidney. Kidney always do reabsorption of water. If there's too much water, it will reabsorb it. So it means osmosis will allow water to be reabsorbed by kidney. Then C, that's colon, talking about intestine. So the colon also always reabsorb water. So remember digestion process. So there must be reabsorption. So how can we differentiate between diffusion and osmosis? In diffusion, I've said that one, it could be gas, liquid, or solid that is moving, but osmosis is only water that is moving. Also, diffusion, that one does not need semi-permeable membrane. What will permit it? It doesn't need that one. But osmosis needs semi-permeable membrane. Then also diffusion, that one can occur it can diffuse into any space. But osmosis must be between two solutions of different osmotic pressure. So it must be two different osmotic content. It means one will be high concentration, the other will be low before osmosis can take place. Now let's look at this experiment. In the experiment of osmosis, so our aim is to demonstrate osmosis. We will be using yam tuber so as our living plant material. It means we can also use non-living, but here we want to use yam as our living plant material, which is the medium, the permeable membrane, so from the yam. So for osmosis to take place, so our materials needed, we need 2 thick slices of yam, we need salt solution that is very strong, that means there is enough salt and little water. So distill water and a trough, which is the container. So method, how are we going to carry out the experiment? We take these 2 slices of yam tubers, we label them A and B, and we try to make holes. We make hole, but before that we have to peel the skin. So it means you don't just use yam that is not peeled. So, and again, it must be raw yam, not the one that is cooked, so that the cell would not have died. So, and you make a large cavity, that's a hole like this in the 2 yams. So then you pour strong solution of salt. That means you put water and salt together, pour it in A to level T1. That means at this level first. But in the second yam, don't put any salt water there, leave it like that. So we call it controlled experiments. So after this, you leave it for some hours and see what will happen. So definitely there will be osmosis. So that's why in the result, after some hours, that one that has salt solution, the level was T1, it will rise to T2. Meaning that because the concentration is very high, salt solution is very high, it will demand water from the distilled water that does not have salt. It will demand. So it means it will be thirsty. Just like someone that is taking too much salt will be thirsty, will be sweating profusely, so demanding for water. So that's why here now there will be what? Movement of water from that distilled water coming into the A. So, and as that takes place, it means there is higher osmotic pressure on the water. That means pressurizing the distilled water, that they need water in this cavity. So the water with lower osmotic concentration will now move through the cells of that yam. So that yam is acting as a semi-permeable membrane, so it will enter the cavity of the yam. So hence there is a rise of the salt solution. It means inside this A there will be rise. But for B, water could not be drawn into the cavity. Why? There is no salt solution, no strong solution that will be demanding water, no pressure on it. So there is a lower osmotic concentration and lower osmotic pressure than the water in the trough. So let's quickly look at a question here. This question is talking about having 4 cells. Maybe you cut yam, cut another thing that are just having cells, and their percentage of salt solution that is in them is 3%. So if you now throw each of them into another solution of salt, that is 4%, 6, 1, 10, and 5. What will happen? The one that is 1, remember, is lower. It means 3% will have more strength than 1. It will be able to drag in that 1. So, and that's why that's the solution that will cause increase in osmotic pressure. It means you will use this osmotic pressure to drain this salt to itself. The water in this salt will drain it to itself. So, and that's why C will be our answer. So that's for 3. Now the next thing we'll be looking at, okay, so here they're saying which of the following yam tissues will osmosis occur. So osmosis will occur in raw and peeled yam. So we said it earlier that you have to peel the yam before osmosis can take place. Another experiment, if we are not using non-living materials, if we are not using our yam, we want to use non-living material like a cellophane. So you can use cellophane, you can use bladder of pig as your semi-permeable membrane. So that cellophane, just use it to cover this funnel. They call it T2 funnel that has a mouth of funnel and tiny long tube. Invert it, so you cover it with that cellophane, then invert it into this beaker containing distilled water. That means low concentration is the distilled water, but the water you will pour into this T2 funnel will be salt water, high concentration. So what will happen to be demanding water from the environment after some hours or minutes. So and that's why the water in this one will begin to rise. But this one, if you pour if you don't put salt water, if you put ordinary water into this T2 funnel, it will not rise, so it will not demand. There is no pressure. So that's just the conclusion of that one. So the next thing we'll be looking at is hemolysis. Once you hear hemo, what is the meaning of hemo? That's blood. But lysis is to break or split, meaning that there will be a splitting of blood. What kind of blood? Red blood cell. Remember, there's something we call plasma. You can use L and P to remember liquid portion. It means in our blood there is a liquid portion containing about 90% of water, so still in the blood. So 90% of water. Others are now food content like the glucose, the amino acid. They are the remaining 10%. So it means if there is too much water in plasma, so it means there is a way red blood cell inside that plasma. So the red blood cell will begin to take the water. It will be absorbing the water, that excess water, and if it keeps absorbing, it will be swelling up until it bursts, and that's what we call hemolysis. But normally this is balanced in our body. That means there is no high concentration or high pressure that will be dragging the water into the red blood cell. That's if the person is normal. So but if the person is not normal, maybe there is, or maybe let's use the word imbalance. If there is an imbalance, that means the plasma, normally the water is supposed to be too much. So but if it is now at the point whereby there is concentration of salt in the plasma now falls. When you say concentration of salt, that means the salt level in that plasma, if it goes down, it means the water will rise, and that's what we call hypotonic. And once the water rises, it will be the red blood cell that is in that plasma will begin to absorb the water and it will burst, and that's hemolysis. So that's what that's entering. You know, the water coming in, we call it endo. Endo is inside, endosmosis. So it's a part of osmosis. Water coming in. So that's endosmosis. So and then, as I said, it will begin to swell until it bursts. So it will swell to the point of turgidity. That means swollen blood, and it will burst. But if the reverse is the case, maybe the concentration of the salt in that plasma is now increasing, maybe the person taking too much salt is not increasing, so it means the water will be low, and that one we call it hypertonic. And anywhere there is hypertonic, that there is too much salt, it means it has to drain water. So it means that it will drain water from the red blood cell. So once it drains water from the red blood cell, what will happen? The red blood cell will shrink. So it means there is exosmosis, it has lost water. Exo is out, it has lost water, so it will begin to shrink, and that's what we call crenation. Crenation means when the red blood cell, when it shrinks as a result of hypertonic solution that has taken water from it, will shrink. So that's crenation. So take note, either the absorption of water, that's endosmosis or exosmosis, both of them can lead to death. In endosmosis, after it has taken too much water, swollen, burst, it means if the blood keep bursting, it means there will be no enough blood. There will be blood shortage, which we call anemia. Han means not, emia means blood. That means there is no blood, enough blood. So if someone begins to ignore that kind of ailments, deaths can take place. So, and for animal cells, they don't have cellulose cell wall, so to resist turgidity, that means absorption of water to the point that it will burst. But assuming it's plant cell, so as it's absorbing the water, it will not burst because of the cellulose cell wall. Now let's quickly answer this question. Mention 3 conditions that may cause hemolysis, that can make blood burst. The first thing is low concentration of solutes in the blood plasma. That means if the blood plasma has low concentration of solutes, it means there is not too much salt. There is only water. That means there is excess of water. That excess of water will be taken by the blood and it will burst. So that's why we say if there is low concentration, it can make hemolysis take place. Then high concentration of solutes in red blood cell. If the red blood cell itself is now the one that the concentration is high, that means there is too much concentration in the red blood cell, but the plasma its own is low, meaning that the one that is high will also still be demanding water. So the red blood cell that is high will be demanding water from the plasma. Then defects. If the red blood cell has issue, so it can also cause demand of water. Then another question here, a red blood cell will hemolyze. It hemolyzes. So when placed in hypotonic, when you put red blood cell in hypotonic, so it means it is absorbing water. Why is it absorbing water? Because cytoplasm is less dense. It doesn't have enough water. It will just be absorbing much water. Then another thing we consider is plasmolysis, turgidity, and flaccidity. What's the meaning of plasmolysis? That means this one will be shrinking. Plasmolysis, that's when there is a shrinkage of vacuole. Vacuole is where cell sap is found in plants. So there will be a shrinkage. So shrinkage of that vacuole. So it means the water in the vacuole, it will be going down. So and it will be shrinking. So and it will even be pulled away. Once there is no water again, they will pull it. It's just like armed robbers. After they have stolen collected money from someone, and then the person does not have money again, and they still kidnap the person. So it means plasmolysis also does the same thing. That means to be shrinking the vacuole, and finally it will pull it away. From where? So pulling away of the cytoplasmic lining. That means there is a cell wall where the cytoplasm is surrounded. Look at this drawing. This thing you see here, it was actually here. That means it surrounds this cell. It surrounds it. But because there is no water, so it has to be what? To be living. Peeling. That means it will be draining, shrinking. So just like maybe one skin, our skin, it covers the whole of our body. So if it gets to a point that the skin begins to peel, so as a result of dryness, so that's what is happening here. So cytoplasm lining from the cell wall. So when the plant cell is placed in more concentrated solution, that's when that happens. So once the cell is in the concentrated environment, that concentrated environment will drain water from the body of that cell, and the vacuole will begin to shrink away from the cell wall. So another thing is water is lost by exosmosis, is losing water. So due to the environment that is high, so vacuole will begin to shrink. See the vacuole. Remember, vacuole is where cell sap is. So having enough water, but the water will be drained, and that's why it will shrink. So the cytoplasmic lining are pulled from the cell wall. So the space between the cell wall and the cytoplasm so becomes filled with concentrated solution. That means the environment that was concentrated, it will now come in. It will come into the to the cell and fill it up. Then the cell becomes flaccid or plasmolyzed. That means that cell that was having water before, there will be no water, everywhere will become dry. So just like a place that's where they have taken operation, maybe all these armed robbers, they carry out their operation, the environment will look tensed. So the same way, there will be flaccidity or plasmolysis. That means the environment become plasmolyzed, no water, flaccid, no water. So we can also call it crenation if it is an animal. So because there is no water again, so crenation. Now plasmolysis can now lead to wilting. That means the plant will begin to wither. That means begin to shed their leaves, or it should die. So that's just what this aspect is talking about. Now on the other hand, when the plasmolyzed cell, the one that has lost its water, assuming it's placed in the water environment that is pure, that there is no salt, so the reverse will be taking place. So water will move into the cell sap by what? Endosmosis. So because cell sap in the vacuole is more concentrated, so the vacuole will increase in volume, and that's why you can see this one is bigger. That means it has swollen, it has absorbed water. And also look at the environment now, it's filled up, so it is not drawn away from the cell wall again because there's enough water. So that's why it can spread. So the cell wall will distend, is spreading until there's no further distension. So and that one we call it turgidity instead of flaccidity. It will become turgid. That means it's soaked with water. Now plasmolysis can lead to death. That means loss of water. It can lead to death of the cell if you allow it for a long time. And that's why if you plant a crop, in a very salty soil, environment that is having high concentration. So that environment that is having high concentration will begin to take the water in that crop, making it to dry, and then before you know it, it will die. Now, what is the importance of turgidity? When plant is turgid, having enough water in its body, so it will make parenchyma happy, because parenchyma, from our last explanation about or last video about parenchyma, that you can use parents to remember parenchyma. Our parents get food and water for us, meaning that parenchyma gets water for plants, so that that plant will be robust, and then to support it, make it to stand firm. So meaning that a turgid plant is favored by parenchyma. Also, lamina of leaf, talking about the leaf blade, you see leaf becoming flat. It's because there is enough water, that's why it's flat. Assuming there is no enough water, it will just be looking wrinkled. So meaning that a turgid plant will make the leaf flat. So because of that turgid mesophyll cell. Turgidity can also help animal cells to retain their shape. Animal that you see that is standing firm, is robust, is because there is enough water. So that's why you can see the big shape of that animal. So turgidity. And all cells, they can carry out their physiological processes. So if they have sufficient water, so when there is enough water, they can run, they can do all their activities. But now let's look at flaccidity. That means this one now is now losing water. Plant that has lost its water. So what do you expect from that plant? So it means it will later die. So, and this normally occur in droughts, when there is no enough water in the environment. So it will make that plant become weak, limp, soft, so they finally drop dead. So there's a drawing here that shows us how turgidity is taking place. Look at the cell from here. So it was swollen, spread its membrane, cell membrane, the vacuole become enlarged. So meaning that this is what the number one. This number one, they say which of the label structure in diagram will shrink when plasmolysis occur. So it means if there's not enough water, this particular one, this number one here, it will shrink. That's the one I showed earlier about the cytoplasm and the vacuole. So how it was shrinking, leaving the body of the cell wall. So that's one. And on the other way around, it will now swell if there is enough water. And that's what the next question is talking about, that the condition shown in the above diagram is due to what? So that's the excess water in structure, the build one. So that means when the water was excess, so this diagram now expands to this point. So that's for the second question. So which of following solution will induce condition shown in diagram? So it means it's hypotonic. If the environment is hypotonic, that means there is no salt there, so it will be absorbing more water, making it to swell up. Now the next thing is Tugor pressure. That means Tugor pressure, pressure of turgidity. What will make something to be swollen, absorbing water. So Tugor pressure occurs in a cell when the volume of its cell sap increases. So once the cell sap is increased, it means it's absorbing water. There is a pressure that is making it to absorb water. Turgidity in the cell of plant does not easily lead to bursting. So why is it that turgidity in plant will not lead to bursting? Because it has a cell wall. That cell wall will prevent it from bursting, even though it has absorbed water. So the last thing for today is active transport. This one has to do with molecules. The molecules could be sodium ion, it could be the amino acid, it could be glucose, all those substances that are being reabsorbed in nephron. So as you can see this example here, so in nephron, when you look at the nephron, there's always a kind of reabsorption, selective reabsorption during urine formation. So how the molecules will now pass across a semi-permeable membrane of a living cell against concentration gradient, that's active transport. So it means this one is just like when someone is climbing a mountain. So that's an active transport. That means you are against the concentration gradient. That means the concentration gradient is high. Then you are coming from a low concentration. So you now force yourself to that high concentration. You are passing through a semi-permeable membrane. So that's active transport. So and at the same time, you are expending energy, expenditure of energy. So using energy, using force to make that transport possible. So just like when these nerves, when it is movement of nerves, there will be the release of sodium ion. That's an active transport. That means forcing itself out. So for movement to take place, just like a vehicle that the tire has been deflated and you are forcing it to move, so it will begin to release some strange sounds. So that's release of those strange sounds represent the molecules that is coming out across the semi-permeable membrane against the concentration. That means it's against the movement of that vehicle that the tire has been deflated. So you are forcing it, moving it. So that's against the concentration gradient. So movement of molecules through semi-permeable membrane of living cell against the concentration gradient. So it will be using its energy. So the example, like I said, selective reabsorption of glucose, amino acid, and sodium in the nephron. You know, in the nephron of kidney, we have something like this. So when the urine will be formed, there are some things that must not go with it. If glucose, for example, should enter into this nephron, so there must be reabsorption. You force it out. So that's an active transport. Also, movement of sodium ions out of the nerve cells of the cause of transmitting nerve impulse. Like I said, when there is a transmission of nerve impulse, that means a message to the brain or anywhere, so it will be releasing sodium ion. What are the factors that can affect active transport? The first thing is temperature. If the temperature is low, it means it will not aid the transport. But if the temperature is high, it will aid it. You know, it's just like when the environment is hot, it will make you to run. So the same way, when the temperature is low, there will be no active transport. Then if a oxygen level is high, then it also aids transport. But if it is low, it will inhibit the transport. So that's where we are going to stop for today. So as we have looked at the topic, the cell and its environment. So thanks for listening.